Hi, can everybody hear me? Yeah. Thank you, Priyanka, and thank you to the organizers for inviting me to the school. And actually, I'm part of the scientific organizing committee too. Just that I'm an outlier. Yeah. I think a good number of you probably are trained in physics and math and stuff. How many of you have had biology in your undergrad? Not bad. Okay, I'm not going to feel so alone. Okay, but I'm just going to try and keep it simple. The idea is to give you an overview of how we in the field think about this problem. How do we understand the processes, the niches, and the mechanisms that allowed for the transition from non-life to life on the early Earth? And in doing so, I'm hoping to give you people a flavor of how can we think about habitability. Thanks to Ravi, a good bit of definitions have been sort of set out in that space, but I'm going to get to that towards the very end because there's a lot to talk about and we shall start at the very beginning. Yeah, you remember, let's start at the very beginning. A very good place to start. Okay, I'm just going to go forward. So this is the bunch of people, and several of them are not on this slide, a whole bunch of undergrads. Together, we try and understand all of the things that I'm going to talk about, not all, a good number of the things that we're going to talk about. And we sort of want to gain a sort of some basic understanding of how did biology come about from prebiotic chemistry, the chemistry that was rampant very early on. Where am I actually starting? I'm starting with this figure. All of you recognize? Yeah. The pale blue dot. When was it taken? Ravi, come on. We were born then. You were not born, so you're excused. Somebody else said 79, you're excused. Feb 14th, you got that right. 1975. Yep. Oh, 1990. Shit. Sorry. I shouldn't have taken that. I'm sorry, I shouldn't have. 1990. I remember the Feb 14th part. Yeah. We were very much born. Yes. But even in 1975. Oh, okay. So the picture tells us all about why we need to take this planet seriously. This is the only home that we know of where we'll survive. I'm not sure there's anything else out there that remotely has given us the sort of understanding that we could survive if we sort of try and populate a new sort of space, either within the solar system or beyond, even particularly within the solar system. And as Carl Sagan very famously said, look again at that dot, which is the size of a pixel in this picture, in the main picture itself. That's here, that's home, that's us. And I'm very sorry on behalf of all of my peers and other people since the Second World War who have screwed it up for all of you and the forthcoming generation. However, it's hopeful, and you people will make sure that it continues as a place that can continue to host life even going forward. And that's the hope that we all live with, right? This field of study of origins of life broadly comes under the umbrella area of science called astrobiology, extremely multidisciplinary and interdisciplinary, and feeds off of various areas of sciences. And this is really not a sort of a complete picture. There's math underlying all of this. There's physics. It's not mentioned out here, but understandings using these diverse tools and approaches allows us to essentially put together the pieces of the puzzle, which allows us to understand how might things have come about very early on. I'm talking about a story which panned out possibly close to 3.8 to 4.2 billion years ago, very soon after the Earth was formed. And here is a statement from Mark Kaufman, who says, by exploring and illuminating the world of extreme life on Earth, what does the extreme life that can exist on Earth now, under very, very extraordinary circumstances where you and I will cease to survive, what does that tell us about the limits of life? What can you understand from the chemical makeup of the cosmos? Why is that important? It's important because if that is the material that seeded the early Earth and led to all the downstream processes, then the chances of finding another planet in our solar system or a moon in our solar system that could have potentially hosted life or can host life is very huge. Or for that matter, even elsewhere in the universe, outside our solar system too. And more recently, a lot of our understanding about this process is allowing us to think more systematically about how do you look for habitability? How do you look for whether a body can be habitable? And some of the immediate lessons that we're gleaning from are coming from missions that have gone to Mars, that have sort of flown by Europa and Titan. And there's a whole bunch of science that's still got to be done. Nevertheless, since 1953, which is the year that the first experiment happened that showed that you can indeed go from a bunch of inorganic molecules to organic molecules that are the foundational molecules or building blocks of life, we have come to understand quite a bit about some of these concepts that I'm going to talk to you about. Students, anybody knows what happened in 1953? Miller-Urey experiment, very nice. But that is the one that set the stage for this field of prebiotic chemistry. What else happened in 1953? The double helical structure of DNA was described, which also played a very important role in one of the hypotheses for the origins of early life, which I will come upon in a couple of slides down the line. But people have been thinking about this not just in the last 80, 90 years, right? You have religious accounts that talk about it across all religions, polytheistic, monotheistic, doesn't matter. The problem, though, is that these are symbolic texts rather than historical accounts, very poignant stories, but mostly of morals and cultural norms. Realism, not necessarily the point, right? Very much centered around how to bring about order in the society, not necessarily meant to be scientific accounts. So hence, doesn't provide a necessarily satisfying explanation, systematic explanation about how both the universe and life came about. But suffice to say, we're not the first ones to be thinking scientifically about the problem. Here is a quote from Aristotle. He wrote this on the generation of animals in book 3, part 11. Animals and plants come into being in earth and liquid because there is water in earth, air in water, and in all air is vital heat, so that in a sense, all things are full of soul. There are words that he's using which is on the non-science side, but please remember that first sentence, and you will see how he wasn't very far from actually how we think about the problem now. And you'll see the connections shortly. Therefore, living things form quickly whenever there is air and vital heat are enclosed in anything. And when they are so enclosed, the corporeal liquids being heated, there arises, as it were, a frothy bubble. Since then, there have been a whole bunch of experiments that have happened. I'm just giving you snapshots of some of the historical milestones. Francisco Redi, John Needham, and Lazzaro Spallanzani first sort of were the scientists who systematically took an approach to understand the theory of spontaneous generation. The idea was that life came from non-life. And in this space, Francisco Redi actually demonstrated that that's not possible because maggots actually are offsprings of flies. So life begets life. Non-life doesn't beget life. John Needham basically argued that microbes arose spontaneously in broth. And life force is a term that many of them used very, very sort of frequently. But Lazzaro Spallanzani actually countered what Needham was showing with a whole bunch of his own experiments. But one of the important sort of things we might have all heard about is omnivivium ex ovo. All life come from egg. And this was proposed by William Harvey, who's an English biologist. But one of the coolest experiments back then itself, we're talking about 1800s, came from Louis Pasteur and John Tyndall. This broth experiment here, this swan-necked flask experiment, they basically showed that in those flasks where you had the swan neck kind of a contraption, it allowed only particle-free air to enter, because of which microbes could not grow in them. But if you did not have this swan neck kind of neck, and if it was just sort of a simple neck, which was open to the external environment, it could very readily lead to the formation of microbes or emergence of microbes. In conclusion, new organisms come only from existing organisms. Yeah. Two very important people, particularly Charles Darwin, whose work has informed our understanding of evolution of species, Not just that, he is also one of those rare scientists who has questioned about the origin of species too. They basically talked about how more complex life generates from simpler life due to random mutations and natural selection. But Charles Darwin particularly questioned, where did the first organism come from? And in a letter in 1871 to Hooker, he wrote this: but if, and oh, what a big if, we could conceive in some warm little pond all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed ready to undergo still more complex changes. At the present day, such matter would be instantly devoured or absorbed, which would not have been the case before living creatures were formed. Do you see some overlap with what Pluto was saying? However, the wording is different a little bit, yeah? More specific, more scientific, using more technical terms. Darwin's, Haji. Yeah, we knew about amino acids before that itself. What we did not know systematically is about the details of their physicochemical properties. We knew that all life forms, plants, animals, microorganisms were all made up of this fundamental molecule, which is probably very important for their sustenance and living. The terminology and the coining of the term proteins and all happened closer to 1840s or 30s. I could be a little off by a few decades, but yes, it was known by then. We knew very important things existed in the cell that were facilitating some of the complex functions. How they went about systematically figuring out key, nucleic acids were the informational molecule, proteins were the catalytic molecule, so on and so forth. The details of it took over a century and stuff, but some of the original experiments started late 1700s. Early 1800s sort of peaked with these experiments. So how then do we think about this problem super scientifically? All you have to do is look at this beautiful continuum that exists between physics, chemistry, and biology. Let's start with biology. Here are two different molecules that I've shown. One is a proteinaceous enzyme. All enzymes are proteins, but not all proteins are enzymes. You remember that statement from school, but that's not the point. This is made up of a biomolecule called amino acid. It's essentially a bunch of amino acids strung together using a covalent bond called a peptide bond. It allows the molecule to fold back on itself and form gorgeous structures, which is super crucial for its function. This on the right side is also an enzyme, but not made up of amino acids. It's made up of RNA monomers. These are called ribozymes, which is where that word comes from. Ribo is from the ribose sugar of RNA. It's actually an RNA strand which can do the exact same thing. It can fall back on itself, form beautiful structures, and can catalyze reactions. But for a second, let's zoom into them. What are they made of? I'm sorry I'm showing only amino acids here because it becomes very complicated to draw out nucleotides, but they are made of simpler monomers, which are made up of what? What are they made up of? Atoms. This is the ball and stick notation of atoms. Black is carbon. Yeah. What is blue? Oxygen. Yeah. There are all of these. White is hydrogen. Yeah. So this is basically saying that it's a bunch of atoms which are covalently linked to each other that forms the monomers. Zoom into that. What do you see? Even more simpler chemical molecules that are very readily even formed in the interstellar medium and the circumstellar medium. So chemistry, rampant. The chemistry that is required for life to come about is just not plausible on terra firma. It's something that's happening in the space. Zoom into that. They're basically a bunch of atoms. So if you can think of this continuum, what it brings us to is a very important theory called the abiogenic theory. Basically, the things you need to remember, living organisms are made up of a finite set of chemical compounds. The chemical compounds all are made up of a bunch of atoms. Can you tell me the atoms? Louder. We all ate. We can break, we can, more energy. Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, CHNOPS. So if you actually look at all of the molecules of life, they're made up of the same set of atoms. You do have minerals present, ever so often you will see metal coordinating some of these important molecules, without which these molecules cannot function. But suffice to say, finite set of chemical compounds, and many of the precursor molecules come together to form higher order assemblies. I'm going to ask you to concentrate on 3 out of the 4 important biomolecular classes of life. This is protein, made up of amino acids. This is DNA and its cousin RNA, both of them made up of nucleotides, a monomer called nucleotides. And these are lipids. Where do you find them? In your cell membrane. All your cells are basically covered by a boundary made up of this molecule. This molecule is a very interesting, the monomer is a very interesting monomer. There's a water-hating group and a water-loving group. So all of these higher order structures are what we think allowed for the transition from chemistry to biology. So in the individual monomer space, none of them can do the function what the higher order polymers can do. They have to become polymeric before they can start doing some of these extremely cool functions that we see in biology. And we call these properties emergent properties because you cannot ascribe them back to their individual monomers. So we think this transition from chemistry to biology or non-life to life was driven by the fact that molecules could form much larger higher order structures, which could then get endowed with these properties that allowed them to do function. So abiogenic theory, very simple. You started with fundamental inorganic molecules shown in this column. They formed precursor molecules, which then led to the building blocks of life molecules. These building blocks of life molecules form much larger higher order assemblies, and all of them get encapsulated in a membrane to form the fundamental unit of all life, which is the cell. So if you then have to now think about transition from chemistry to biology, how can you structure it? I've given you the pathway already. You need to understand every single transition. That's one intuitive way of thinking about it, which is what the abiogenic theory is. And given that there were several hundred millions of years, right conditions, you can very readily sort of envisage that these kind of transitions were plausible. Haji. So this is something, so you sort of, the question is, do we know if the molecules came before the lipid layer? So this is something that you're sort of alluding to what is called in the field the lipid world hypothesis. And the scientists that proposed that basically said that amphiphilic molecules, which are very readily found even in, say, meteorites and comets and stuff, asteroids, essentially catalyzed a lot of important reactions, which then formed the other molecules. But fundamentally, nobody questions the fact that all of these were there. Depending on your favorite hypothesis in the field, people have differences of opinion about this. I, for one, think a very systems approach is required because they were all simultaneously present in the prebiotic soup and nobody gets a bigger advantage than the other. But what that leads to is a problem which basically complicates the kind of complexity you're dealing with even in the molecular space, that you can't track reactions systematically, because of which people have taken very puritanical approaches in the last 60, 70 years. But in the last 15 years, a bunch of us have made lives hell for ourselves, and we're trying to take this more holistic approach, which has its positives and negatives. I'll touch upon it when if I get a chance, but what you're saying is the lipid world hypothesis, and I'll get to it in a second, but this allows me to, Haji, Jayesh. Very important, very important. So turns out certain reactions can happen without metals or any catalysis required, but a good number of these reactions at every step were possibly driven even more effectively in the presence of metals. No, it's not at all. No, it's not at all an exhaustive list. This is a consensus list. And the consensus list is bigger than this. It's just that this is a cartoon that I found that I could put here. The consensus list is bigger. And typically a prebiotic chemist way of thinking is that mujhe agar kuch banana hai, if I have to make something, I'm going to think about what the fundamental molecules could be by looking at the structures, and I will find a way to justify its prebiotic plausibility. But the best way to do this is to probably look at the catalog that the astrochemists have generated and the people that have systematically tried to understand terrestrial syntheses over the last several decades. Unfortunately, no such catalog exists. It's strewn all over the place. And ideal would be to have all of these molecules present in your reaction sort of background to be able to understand the so-called holistic thing that I'm talking about. But it gets very complicated very quick, which is why people sort of have focused. The historical method has been to focus on individual groups of biomolecules to understand how they might have come about, to at least sort of tease the steps, the pathways, so that you can think about how all of them could have come together to assemble the cell. But there's no reason to believe that this all happened in isolation. Not at all. No, go ahead. No. One of the examples is what Ravi touched upon, and there is a recent report from 2025, the papers in bioRxiv. People have figured out an abiotic synthesis of DMS in interstellar medium. So I'm not sure. So this goes to a broader problem in the field. Is there a consensus about biosignatures? He's the boss, but I can tell you that there is, at least from where I'm coming from, understanding these processes, I feel like the so-called biosignature list has to be revamped, and you need to introduce pre-biosignatures in there, because that is probably something we're not even looking for, because we are right now wearing blinders. And there's a whole bunch of things, in my opinion, to understand if we actually find these pre-biosignatures, because that might be telling us that, hey, here's a body where life is probably on its way to emerge, given the right conditions, given all of the other conditions. So one example would be sugar phosphate backbones. These are not nucleic acids, but these are fundamentally the backbones on which your nucleic acid is made, and they form very readily. These are. But the other side, which I would like to present a counter for, is they're very polar. There's a ton of hydroxyl groups on it, and they're very, very soluble. So they don't have to form, clumpy structures and crash out like proteins would, especially proteins which have lots of hydrophobic residues can form these beta sheet-like structures and crash out. But these things don't necessarily have to crash out. But the other problem is, under very high temperatures, which is probably what is happening in early Earth-like conditions, they form intangible products. It's called advanced glycosylation products, which make their detection a pain in the wrong place. So that is a problem. That's a very big problem. We can talk about it. I'll have to think about it a little bit. But this is we wrote a very sort of a preliminary sort of perspective piece in Life about this. I truly feel like the biosignature space can undergo a little bit of revamping, where a bigger number of molecules are also accounted for, which can be considered from a habitability context. We'll talk about it a little more. Yeah, sure. Haji. They can very readily form, yes. Are they formed in modern bio? Do you see them in extant biology? Is that the way to? Like Charles Darwin said in that thingy, if it's a source of energy, so they would get devoured like that. It's basically sugar phosphates. So sugar is one of the most important energy source. So even if they're forming, they're very readily getting eaten up. I'll show you structures of that when we get to that point. Haji. Yes, very much. They don't have to all be, yeah. They could be spread out. If they're too spread out, then the chances of the products meeting each other to lead to more interesting higher order complex molecules is more difficult. But if we can think of environments where you have, say, multiple sort of hot spring conditions or terrestrial geothermal pool conditions, where different pools maintained at different pHs with a different set of cations and anions in them because of the mineralogy that surrounds the pool, allow for different kinds of reactions, but interconnected, waterways or whatever. They can bring these molecules together to a third space where their crosstalk becomes more amenable. And there are reasons to think that this is very plausible, because there are modern terrestrial geothermal spring systems which still have these kinds of variety just within a few meters apart. And we work with some of them actually in New Zealand in that area. But all of this segues to some very sort of I just want to give you a flavor of the hypotheses that are out there. The primordial soup hypothesis is the most intuitive. Yeah. So you had a primordial soup where you had all the ingredients required, all those precursor molecules very readily available. Either they were delivered via exogenous delivery mechanisms, asteroids, comets, meteorites, so on and so forth, or you could even have terrestrial syntheses of this that could then populate this pool from where other interesting reactions can happen and more recognizable biomolecules could have come about. Now, this is a terrestrial system, and what is unique about the system, people? It's almost always at the edge of a volcano. So what are the energy sources that are possible? Geothermal energy. The other one is just right there. Radiations, all kinds of radiations, including lightning, UV radiation, name it, ionizing radiations. Another small group in the field talk about this hypothesis, which is basically the deep sea hydrothermal vent systems. Again, volcanic systems driven completely by geothermal energy, no light because it's several kilometers down on the ocean floor. However, there's a lot of very interesting mineralogy happening here, which actually makes these environments excellent for redox chemistries. So in that sense, I will not write it off fully, though I find it more difficult to wrap my own personal head around this hypothesis. I'm more sort of aligned with that mainly because it's more tangible for me. But wherever this happened, it goes back to the question that one of you asked. I forget. Minerals, Jayesh. All these things, chimneys, all these pool edges are lined by very interesting minerals, and rock water chemistries allows for leaching out of ions into these spaces, and they're ready now for doing interesting things with these other precursor molecules present in the vicinity. So clay hypothesis is something that was separately proposed by a gentleman in the field, but I feel like this, whatever is your favorite sort of hypothesis, you can still think about role of clay and other minerals in these processes. But I will bring your attention to two main sort of arch rival hypotheses in the field. RNA world hypothesis, very, very famous, has a lot of proof in extant biology and also experimentally speaking. So question for the non-biology audience, and then I'll get to the biologists and biology undergrads in the crowd. Do you know what is the core reaction that allows you and me to be alive? It's okay, I'm not going to know, you're not going to lose any marks if you say wrong. Sorry? Into energy? What is breaking glucose? Who said respiration? Very important. How is respiration possible? What makes respiration possible? Fundamentally, his breaking of glucose and your respiration is connected by something. Enzymes, proteins. Yeah? No proteins? Death. Period. Respiration involves a very important molecule called hemoglobin, a very complex protein which has 4 subunits. Your breaking up of glucose has a whole bunch of enzymes. It's like the Krebs cycle, right? It's a whole bunch of enzymes. Root molecule, proteins. So what is the actual reaction that keeps you and me alive? Okay, now the biologist can speak. The fundamental, he kind of almost said it, the fundamental reaction that makes enzymes or proteins in your body using the information in the DNA, which is your ribosome, the reaction which happens in the ribosome. It's a peptidyl transfer reaction, which essentially allows amino acids to join together to form proteins, large molecules. That is what allows us to survive. And why did I even start saying that, paper? Shit, I forgot. Anyways, RNA world hypothesis posits that information coding and this very important catalysis that is done by proteins can actually be done by one single molecule called RNA. Unlike DNA, which only encodes information in our cells, RNA processes information, does chemical catalysis, does a lot of amazing things. So just maybe one easy way to think about emergence of that one biomolecule which did both the functions is to think about how did RNA molecule come about on the early Earth. So this one has a lot of proof both in biology Now I know where I came from. Biology, proof in biology, both in biology, current biology, and also abiotic reactions. Yeah. So very nice. She's raising two very important points. She says RNA is an unstable molecule because it's very prone to a reaction called hydrolysis. A lot of water, right? All of this is happening in the backdrop of water. Like Ravi pointed out, solvent of life, water. No water, no life. Then how do you think about both these problems, hydrolysis problem and she raises a concern called the mutation rate. What happens is when these molecules form, even in our cells, when the molecular information is replicated, there are chances for mutations, which are basically issues when information is copied incorrectly. When that happens, a whole suite of molecular machines called enzymes will come and correct for it. If they don't correct for it, it leads to disease. What happens on the early Earth, this is veering away from the chicken and egg problem. Did proteins, you have to make proteins, you need nucleic acids, but the nucleic acids are stitched up by proteins, which came first, which is where the RNA world hypothesis provides a wonderful respite. And in this RNA world hypothesis, we don't care too much about the higher mutation rate. The reason being, if you all agree that RNA world was an important stopgap prior to the emergence of modern biomolecules as we know, and I'll show you that early history of timeline that we think about how things happen on Earth. Mutations are good. Too much mutation destroys information. But mutation at some level allows you to sample a whole bunch of interesting molecules much quicker. So on the early Earth, RNA having a higher mutation rate was not big of a problem. In modern biology, RNA having mutations, serious problem. Yeah. The other opposite group, for the longest time when we went to Goddard research conferences, which are these very small group conferences that happens in the US, where the who's who of the field are there, there used to be like battles between people in these two groups. This is a minority group, which is again gaining traction in the field, this hypothesis. Metabolism first hypothesis, people say, RNA is such a complicated molecule, not readily possible in your Urey-Miller kind of reactions. How do you even make it? So you first probably had networks of small molecules that could readily assemble to form, Krebs cycle-like processes, which allowed to make some of these monomers, which then got polymerized into your nucleic acids. So there is still no full respite because going all the way from the prebiotic soup to a fully self-replicating RNA is non-trivial. And there hasn't been a single minimal metabolic network that has been described to date that can very readily form all the fundamental ingredient precursor molecules of life. So we still do not have a recourse. People working in both the fields are still doing what they want to do with their favorite molecules. So this takes me to the early history of life on Earth, as we think this is sort of a simplistic one, enough for this audience to get a sense of what is the timelines we're looking at when all of these processes happened. All of the interesting prebiotic chemistry was happening very readily, literally 500 million years after the Earth formed. How did you go from the prebiotic soup space to an RNA world space, and how long did it take? There's still no consensus about it. But I will tell you this. The oldest signature of life that exists to date is from 3.95 billion years ago. Yeah, 3.95 billion years ago, very close to 4 billion years. And this is a real signature, and I'll give you a flavor of what it looks like. But we try and understand the processes we are interested in understanding roughly in this time space. And we are, again, in my lab, we're not married to the RNA world hypothesis. We take a much more sort of holistic sort of an approach and delve into more messy chemistry spaces. But this is a reasonable way to think about how do you go from simple molecules to more complex molecules. There are a few things which are a given. And for this crowd, I'm not going to really delve into it. But when did the solar system form? Consensus. When did the moon form and how did it form? The Thea, the whole giant impact hypothesis, which involves a Mars-sized object called Thea. Consensus. Late heavy bombardment, depending on whom you talk to in the field, this is probably debated. But even if there wasn't late heavy bombardment, there was enough of bombardment to bring in a lot of exogenous material from outside of Earth onto early Earth. It happens even to date. I'll show you some examples from art, at least, I mean, yeah, we were not around necessarily when it happened, but we worked with those samples. This exogenous delivery is thought to have brought in a lot of interesting material in addition to the chemistry that's possible on terrestrial environments. This is debated. When did the Earth's crust and oceans form? Maybe there's more consensus now. But one thing is clear, we need a crust and we also need water. No water, no life. At least not how we understand. So given all of these prerequisites, the most important next step is to come up with a definition for life. Google definition for life and you'll get thrown up a few hundreds of answers, right from the spiritual to the loonies to the crazy stuff or scientific all over the place. So what NASA Astrobiology Institute did is to constitute a working group that was led by a molecular biologist called Jerry Joyce, but the group was very diverse. It had astrochemists, planetary scientists, geologists, molecular biologists, prebiotic chemists, all of them working together to come up with a working definition that we use when we do our experiments. Basically, you're looking at a slide which says there are 3 important hallmarks you have to consider when you're thinking about living entities. It should be able to, once formed, once it came about, it should be able to propagate itself, replication, and replication is possible only when you have information. Without information, it's not very easy to replicate something, right? Metabolism, which allowed the systems, living entities, to exist far from equilibrium, because equilibrium is death. If you ever make that t-shirt, you give me the copyrights. Equilibrium is death. The third very important thing, which I will get to towards the second mid part of this talk, is the importance of compartmentalization, forming boundaries. Again, no cells, no life, no water, no life, equilibrium is death. Yeah? 3 important things that you can remember. Haji. Absolutely. No noise. Okay, the no noise, the other way to think about this is bringing molecules that are important together to be able to talk to each other. If you can't envisage scenarios where they could come together and do something, forming them would have been pretty useless. You could have gotten lost, completely useless. And the reason for compartmentalization being articulated as compartmentalization will happen very quickly, and you'll see that. Just give me a minute and I'll get to that slide. You said something. Very nice. That'll be the word, homeostasis. I'll be fine with that, yes. So metabolism is energy, self-replication is information, compartmentalization is homeostasis. You need to think about fundamentally these 3 parameters when you're thinking about concocting lifelike entities using a bottom-up approach in the lab. So where did the fundamental precursor molecules come about? Exogenous delivery, terrestrial synthesis, in environments basically that allow for these reactions to be possible. What do I mean by that? Look at all the biomolecules. Excepting for lipids, the other 3 classes of biomolecules, which are proteins, nucleic acids, what's the third one? Which I'm ignoring completely, but it's there. Carbohydrates. What are these? These are polymers. And how are they strung together? If you think about a chain of pearls, each pearl is a monomer. The string that holds this polymer together is essentially covalent linkages, which stitches up the individual monomers to form a backbone. So your proteins are connected to each other. Amino acids in the proteins are connected to each other by a covalently linked covalent bond called peptide bond. Nucleic acids may, the bond is called phosphodiester bond. Sugars may, in carbohydrates, it's called glycosidic bond. But fundamentally, what is this bond an outcome of? It's an outcome of a reaction called a condensation reaction. How many of you have heard the term condensation reaction? It's a chemistry term. 11th, 12th grade. दो molecules को बगल में लाओ, kick a water molecule out and form a bond. Literally, that's what it means. Now, all of the drama is playing out in water. And you have to kick a water molecule out for making every single bond. So you understand how a high-cost reaction this is. Basically, these molecules love to be single, don't like to mingle. You force them to do that, and then you form these biopolymers, and only then do you get life. So you have to keep in mind that when you think about these processes abiotically, no proteins, no high energy ATP molecule that allows the proteins to catalyze all of these reactions like in our cells. What environments would have allowed for these high energy uphill reactions to happen? And I'll show you some ways of thinking about it. So this is just to say that the prebiotic milieu had all these interesting molecules. Now I'm beginning to show a little bit of structures. Yeah. So here's your nucleobase. This is the informational moiety in your nucleotide, which allows information transfer between nucleic acids. Here is your amino acid. Here is your lipid monomer. There are enough number of chemical pathways that have been described, plausibly prebiotic, can be justified, that allows for their presence on the early earth in the prebiotic soup. So then the question is, this heterogeneous soup, which had all of these molecules, would have also had to, in certain niches of the, in certain spaces of this niche, facilitate interactions which led to the emergence and evolution of these molecules into the polymers that we saw today. The exogenous delivery bit. Here is a proof. This particular meteorite fell in 1969 in Victoria in Australia. Victoria is a, like a state. Yeah, Victoria is a state in Australia. It fell in a village called Murchison. This is one of those first carbonaceous meteorite that has taught us a lot about the chemistry that would have been plausible in interstellar medium that rode on these bodies and got delivered onto Earth. I don't know how much you can see that, but that's all the molecules that have been demonstrated in these rocks, in these chondroids. Yeah. Since then, there have been other chondroids that fell, and the story repeats itself. This is Murchison meteorite, but there's been Murray meteorite, which has also taught us a lot about the chemistry that's possible in the interstellar medium that can then ride on these entities during planet formation or early solar system formation, and sort of they stay as asteroids and then they kind of get delivered onto Earth via other means. This is a very recent example. How many of you have heard the word Ryugu? The ISRO person raised the hand first. Ryugu is a small piece of an asteroid that was basically Hayabusa 2 picked up Ryugu and brought it back to Earth. This is a JAXA mission. And they systematically looked at what is the chemical makeup of this sort of rock. And they found all of these molecules. And all of this is in biology today. Amino acids, carboxylic acids, like lactic acid, a lot of formic acid, a lot of them. Amines, heterocycles, like the kind that your informational molecule is made of, polycyclic aromatic hydrocarbons in huge amounts. Along with Ryugu, there was a US mission also, which 3 years ago described a whole bunch of these molecules and others. Anybody know what was the name of that rock? Osiris Rex, Bennu. So I'll get to that, but this comes to the compartmentalization thing. On the slide, you have a whole bunch of living entities, right? That's Nelson Mandela, T-Rex, single-celled organism, a giant catfish, a giant redwood, a mushroom. What do you think is the least common denominator of all of these entities? Ta-da! So if you want to think about how you want to make the first living entity on life, all you have to think about is how do you go from the fundamental ingredients and make it into a cell that is bound by a membrane where molecules can talk to each other, do interesting things, undergo replication, grow, divide, and even experience a version of primordial chemical evolution. So this is how we think about this problem. We know for sure that this exists. This is an example of cyanobacteria, one of the first prokaryotes, which I told you, right? 3.95 billion years ago, you have proof. This is that particular entity is the ancestor of these molecules. This is called Glyocapsa. But very early on, prior to that, you probably had something much, much, much more simpler. You had simplistic primordial cellular entities. These are called protocells, which would have done a lot of the things like your modern cells, albeit in a much more crasser and inefficient manner, but very much did the same things. Took energy from the environment, propelled the metabolic reactions in their systems using small networks of molecules that produce the energy that they could use, grow, divide, and sort of, undergo multiple cycles of this, talk to each other, do a whole bunch of things. Because we think that at some stage you had a common ancestral community of cells before. How many of you recognize the tree of life in biology? This is a very cartoon representation of that. This is to say that the modern 3 domains of life, if you take all the genes back that are present in these organisms, you can go back to a base of the tree, which biologists think was populated by an entity called the last universal common ancestor. And this common ancestor harbored a minimal set of genes which allowed it to survive and thrive. But due to certain selection pressures, at some point it divided into the 3 branches of life. This is the historical sort of notion of the tree of life, but a more realistic picture is here. It probably was not one Luca. There's no reason for me to at least imagine that it could have been just one Luca. Maybe it was a common ancestral community of primordial cells, undergoing a lot of fun crosstalk, affecting each other's presence in some ways, undergoing evolution. We actually in the lab have shown that you can have evolutionary processes, prey-predator-like dynamics exist in the space too, where there's no protein, no genetics, nothing, simply driven by the physicochemical properties of these entities. But suffice to say, this bottleneck eventually allowed for the 3 domains of life to come about. But this is to tell you how even the most simplistic of all cellular life forms on Earth today looks. Here's the example of your prokaryote. What stands out? I showed you a tennis ball and I said that's a protocell. What does this look like? Did you say tetanus? Tentacles. It's bloody complicated. Does that scream at you? Even the most simplest of all life on Earth today is very complex. But it is way more simple than our ancestral cell. This is a eukaryotic cell. This is what we are all made of. This cell being so simple and so, underappreciated, basically it brings us all down to our knees for very many different reasons, can do something which you and I cannot do. They can exist at the edge of life. All crazy conditions, extremely high temperature, extreme high UV radiation, extreme high content of salt in the environment, very low temperatures. They will survive, you and I will die. Even the smallest of more complex organisms will not survive these conditions, but these single-celled organisms do survive. So a subgroup of astrobiologists in the field try and understand, okay, if we're now sending, if there are a whole bunch of gadgets on Perseverance rover that's hanging around in Mars right now and looking for samples, what is it looking for? Not the kind of cognitive beings that Ravi would like to find. The kind of entities that are plausible, readily plausible, which is microbial, which is very simple, very, very simple. So we can understand better way to look for them, how to look for them if we actually also know better how to appreciate how these single-cell organisms survive these very harsh conditions. So this is the other approach. It's called the top-down approach. This is the proof, people. 3.95 billion years old wala proof. Na? This is not the exact set of stones from which this proof came, but this is the same kind of entities which result from a crosstalk between geology and biology. These are called stromatolites. These are pillow-like structures. They are formed because of a crosstalk between sedimentation and cyanobacterial sort of interactions. These are dead. There are living stromatolites too. This is in the western part of Australia. And if you take one of these stone-like structures and slice them up, you basically see these striations. And this shows this, you can extract material from here and look for isotopes, which tell us whether it's biological or not. And the first cyanobacterial signatures come from this and other hot spring deposits in the Pilbara. Something very important happened after cyanobacteria came up. So you remember the slide that Ravi showed about evolution of gases over time? What did he show? When it was 4.56 billion years ago, what was there? Carbon dioxide, monoxide, methane. When did oxygen come about? That is because of these guys. They do something super important. Can anybody tell me what process that is? It happens even today, which is where we get our oxygen from. Absolutely. Cyanobacteria are anaerobic organisms which basically take up carbon dioxide and water from the atmosphere, chomp, 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 make their ATP molecules, spit out oxygen as a byproduct. So very early on, maybe when oxygen first came about, a whole bunch of organisms that didn't know how to live in the presence of oxygen died out. But those that figure out ways to survive populated the planet, and then the rest of the things take off, right? Oxygen, super important for more complex life to emerge. I completely concur, no questions. But I feel like one thing where I slightly will make you rethink about where complexity emerged much early on, like almost Cambrian explosion ke time pe. That is when the greatest complexity of all life came about. We are an example of highly engineered complexity. We, elephants, dolphins, super, super, super high complex. But yes, no oxygen, the chances of coming this far, very difficult. These are ancestors. So if I put the evolution of life on Earth, linear wala line into a clock-shaped thingy here, roughly around afternoon is when our ancestors came about, and this is 2.5 billion years ago. 2.5 billion years ago. What did they do to the planet? This is what? This is an artistic rendition, okay? This is what your planet is supposed to have, our planet, not your planet. Sorry, I still live here. This is what our planet used to look like back then. When life happens, what happens? It becomes the pale blue dot that Carl Sagan talked about. So when you are looking at so-called biosignatures and everything else, you need to also appreciate that the planetary history and the geological history of that particular entity is supposed to be taken in conjunction with the signatures that you're looking at. So biosignatures need to be on global scales for us to take them seriously. But I'm jumping ahead. I'm just going to go back to my favorite thing, which is to understand this transition. I'll give you two examples. I think I have convinced you that the precursor molecules are possible. A whole bunch of different kinds of chemistries allowed for them to come about. Atmospheric synthesis, when all these precursor molecules are present, there's a whole suite of molecules that can form. Extraterrestrial delivery, I've given you solid proof. There's a whole bunch of interfacial chemistry that is crucial for forming more complex molecules from simple molecules because surfaces bring monomers together. If you are thinking just about water, where everything is happening in a three-dimensional space, the chances of collisions are very rare. But provide a surface, and suddenly molecules will come. If the surface is conducive for constraining them via certain interactions, they all come together, hang out much closer. So several different interactions are possible. A lot of very cool interfacial chemistry has been shown to be responsible for facilitating formation of the very uphill reaction kinetic thermodynamics polymers readily under wet-dry cycling conditions, which are very common geological cycles driven by day-night cycles, seasonal changes, so on and so forth, happening today on Earth, happened even on our early Earth, and a whole bunch of hydrothermal synthesis possible, right? So what did it do? It allowed for the formation of prebiotic soup with a rich number of primordial molecules. When did all of this actually come about? It came about as a theory, much, much, much before even, I don't know. Yeah, really very early on, 1940s. These two gentlemen separately sitting in two different parts of the world, Russia, American with an Indian connection, Operan and Haldane, proposed the exact same hypothesis in slightly different words. 4 years apart roughly, they hadn't met each other. So 4 साल में उनको पता भी नहीं था कि एक बंदे ने already proposed कर दिया. When it came to be discussed, people realized that both of them were saying the same thing, but they were saying that if you have fundamental ingredients and energy source available, you can actually go from a bunch of inorganic molecules to a whole bunch of organic molecules relevant to life biochemistry. A theory is a theory till your experiment demonstrates it. No, you just need very high energy. So it was simulated in the experiment using tungsten electrodes, but it could be ionizing radiations, anything that can provide a lot of energy to get over this delta G, because you need to bring molecules together to form this condensation-driven bonds. There was probably a lot of light. It was very hot. So in addition to your ocean itself being a boiling sort of pot which kept undergoing a lot of evaporative cycles. There was also a lot of this lightning happening because of, I'm sure they can tell, you people can tell me better why that was possible, but there's a notion that this was the case. But you also have ionizing radiations, UV sources of energy. Collectively, all of them would have provided enough energy. More spurts. Yeah, in spurts. Haji. Pathways about how they happen. So I don't know how the condensation reaction problem exactly works in the ISM conditions. ISM is heavy vacuum. Correct me if I'm wrong. Yeah. Pressures, very low pressures, right? So what exactly drives them to form in so if you take the Murchison meteorite, that doesn't have just our 20 amino acids, which biology uses now. It has a whole bunch of other amino acids too. What are the exact physicochemical processes that drive them? I'm not sure, but they're very readily formed in interstellar medium conditions. Because there's a lot of high energy, a lot of atoms present, nuclear synthesis going on, but go ahead. Oh, no. So, no, here's the problem, right? Every time you have exogenous delivery is also coming at a price. It's coming at a price of impact. I did something. Impact. What does impact do? Resets everything. So, leke bhi aaya hoga, populate kiya hoga, but the next big impact can reset the whole business back to T is equal to 0. So, it is very important to think about both synthesis on terra firma and also the exogenous delivery. I feel like you lose out by ignoring one versus the other. Both pathways very important. This way, nobody can say, where are your fundamental molecules coming from? Very readily possible from two sources. And depending on what was contributing more, the source pool always had molecules that could get replenished, which is very, very important, especially if you're looking at timelines which are very large. Sourav, I'll come to you in a second. No. Possible. However, India and USA were not that far apart very early on. We were one giant landmass. So the ripple effect is very huge. These are, no, actually you need continental landmasses for some of these reactions to happen. So the terrestrial geothermal pools that I'm talking about, and this is something that a very dear planetary scientist friend of mine, Ramon Brasser, maybe you know him, so I'm not sure. He tells me that small enough landmasses would have been readily feasible. You're not probably going to see continents, but small landmasses where interesting intertidal chemistry or volcanic geothermal kind of chemistry could have been facilitated. However, resets are a real thing. There was the late Venier event, which is supposed to have happened with a Moneta-like impact. If that kind of an impact happens, it can reset the geochronology a little bit. Where if the geochronometers are getting reset, then the molecules are getting lost, in my opinion. So I feel like it's worthwhile to think about both these contributions, both from exogenous systems and from terrestrial. But the exogenous thing, one is super important in one context. If you're thinking about habitability, if that's possible, if this chemistry is possible there, all you need is to find a planet or a moon, excusez-moi, that can host the kind of conditions that allow for other downstream reactions to happen that could lead to the transition from non-living to living. So that makes the whole, this goes back to the Drake equation that you were talking about. If at least the fundamental precursor molecules are so prevalent, and given reasonably common geological histories, I don't think they're exactly the same, but if it's possible, depending on how far away are they from their sun, and if their star, and if their star is very close to our sun, the kinds of chemistries that are possible is reasonably similar. But I think there's a whole bunch of other planetary formation history and geological contributions that can determine whether certain things can take hold versus not. So maybe that is what is missing right now in our understanding when it comes to the habitability related issue. Yeah. Oh, I see. Made in the protoplanetary disk and getting a... So there are two... So that is one of the ways that at least Sagan and Scheiber have hypothesized where all of these very interesting carbon-based chemistry could have been happening. And I'm actually going to that next. But a theory is a theory is a theory till it's proven with an experiment. The two important niches that most of the people in the field work with, either it's a volcanic system on the ocean floor or volcanic system on terra firma, for various reasons that I've talked to you about. What am I coming to? I have 23 minutes. Maybe I just... So this is an artistic rendition of... Maybe, Shyama, this helps with your question. So you see how the pools are all interconnected? Different pools can have different local conditions, which allow for certain reactions to be plausible. But when they kind of, come through these pathways to join this mainstream and go into newer spaces, where other kinds of chemistries can be more readily facilitated, one can envisage a scenario where different reactions that prefer different conditions and different ingredients can still be possible, and a third new set of molecule can come about. It's feasible. It's not an unreasonable way to think about this, because we have examples on our planet right now. So here is this model for the terrestrial origins of life, which posits that if you have, this is a tidal zone region, but it could be a volcanic geothermal pool, no problem. If you have the starting precursor molecules, and here is an example of the membrane monomer that we talked about. Our cells are made up of very... Haji. Oh, panspermia. But it is that. I think no, we don't discuss it in literature anymore. I don't see too many people that throw their weight behind this for the singular problem that we don't even understand, given all the aspects we know about the history of our ancient Earth and so on and so forth. We still haven't been able to reconstitute all the steps that led to, all the way from t is equal to 0 to what we see today, or what we saw 3.95 billion years ago. So to transport this problem to another body, of which we have no freaking clue of, I feel like it's just sending the problem somewhere else for which we'll never find an answer. But the other important thing to keep in mind, a lot of these living entities will not survive re-entry processes. Life will not survive re-entry process. Non-living molecules can survive re-entry process, no problem. That is why exogenous delivery is very readily tangible, but I'm not sure if microbes that are really small and that survive at the core, to date, we haven't found them. The Murchison meteorite, the Murray meteorite, there's been one more that fell in South America. We didn't find any bacterial proof. Possible. But this is all we know, Manoj, and this is why we can think a little bit more systematically about this. So Mars, for example, like Ravi pointed out, is thought to have harbored life very early on in its history. The microbial life that we see today, it could have been cousins of the same. I mean, it could have been very similar, but unless and until we find some concrete proof of some nucleic acid or some remnant of nucleic acids there, we'll never be able to say this beyond doubt. But to go back, panspermia, not necessarily considered as a viable hypothesis in the field anymore, not to my knowledge. But if you have the right ingredients, and very readily plausible because of hopefully all the proof that I already gave you. So if you have the molecule here, I don't know if you can see these red squiggles. These are supposed to be the monomers of your informational or catalytic polymers. And these white molecules are basically your monomers of the amphiphiles or the compartment. If they're present in these niches on the early Earth, geologically driven wet-dry cycles very readily allow for the formation of polymers over every dehydration phase. And these two are real images. This is a freeze fracture image of how many of you have heard the term vesicle? These compartments have a specific term. They're called vesicles. When you take lipids, put them in water, they ball up into these compartments called vesicles. When you dehydrate, when you subject them to dehydration, they form these multilamellar sandwiches. And if you have polar molecules in the vicinity in the pool, they very readily get constrained in every alternate layer, which has the water-loving head group present. What this is doing is bringing the molecules in close enough proximity for them to undergo interactions. And it's also reducing water activity, which is facilitating the uphill condensation reaction. In the next rehydration phase, they can sort of get encapsulated in these bubbles again, and these bubbles are these protocellular entities that I'm talking about. And here is actually the image of a synthetic lipid in the lab into which we can very readily encapsulate polar molecules. The dye we used is actually a water-loving dye. So this is sort of the model which says that multiple cycles over several hundreds of millions of years could have very readily led to the formation of a whole bunch of ancestral community of protocells that could have talked to each other and undergone even more complex reactions to result in life that we appreciate now as life. It involved a whole bunch of these steps. And I talked to you a little bit about the prebiotic chemistry. I didn't tell you how we think about the oligomerization, but once you have monomers, you need to think about pathways in which, without any enzymes or any complex biochemistry that exists today, how do you make the string of pearls? Where dry cycles, like I just showed you a slide ago, very possible. And it's thought that once you have enough molecules of reasonable length in the space, some of them could have organically, quote unquote, naturally evolved functions, which then you need to think about how this important information for function got propagated, again, in the absence of enzymes, and there are ways that we think about it. But suffice to say that if you could get to a space where you can form RNA world molecules, and there were other pathways that allowed for early protoenzymes to come about, which are based on your amino acid and peptide moieties, the crosstalk between this catalytic moiety, the informational moiety, and the primordial membranes could, in principle, have allowed for the formation of the earliest cells on Earth, the protocells. I just want to leave you with one thought. This is just a cartoon which shows the same. Whole bunch of monomers lose water, can form polymers. It's thought that from a mixture of these polymers, there were some selections, there were some processes which you can think of in many different ways: availability, stability, reactivity, that allowed for the selection of certain molecules that then evolved into what we see as important biomolecules today. So what this means is that the question of life's origin is essentially a problem of information transfer from a geological environment, geochemical environment, to a highly localized volume. So if you want to think about how this transition would have happened, you need to worry about the prebiotic inventory of molecules, or at least know what they were. Right now, we wear blinders. We look at biochemistry, we see what molecules there are, and we walk our way back and say, oh, this is the monomer that I want to work with. But then this space would have been super vast. Earth had oceans, rocks, minerals, and a whole suite of small organic molecules, essentially allowing for the basic structure to exist that allow us to think about this process. I'm only left with 15 minutes and a lot of slides, so I'm going to think about what I want to talk to you about. But one thing to sort of keep in mind is when you're thinking about these transitions, you need to keep in mind that when you want to think about life's origins as a problem in emergent chemical complexification, what are the Hadean geochemical environments that would have allowed for these processes to be feasible, and what might be the features of these environments that would have allowed for these processes to occur. And because we have discussed many of these things in various forms, I'm going to sort of skip through the next few slides, which just go into the details of these processes. And I'll just get to, I'll probably just talk about this and I'll finish it off, because he talked quite a bit about the habitability part. So this is from Chaiba and Sagan's paper, where they show, actually they did a pen and paper calculation, a very logical approach to figure out how much of carbon could have been delivered to Earth. And this is roughly 10 raised to 9 kgs, the calculation, yeah, comes to. Carbon per year is getting delivered to Earth particularly got delivered to Earth during the late heavy bombardment period, 3.9 to 4.5 billion years ago. Late heavy bombardment as a concept is being debated now, but suffice to say that even if it was not one concentrated period in time, in over several hundreds of millions of years, we would have had enough of carbons coming onto Earth. Why is carbon so important? All of life is carbon-based. Do we know of another molecule that can replace carbon? It cannot replace, but I will tell you why. These are the complex carbon molecules that can even form in the interstellar medium. These are called polycyclic aromatic hydrocarbons. These structures are very readily detectable via various, these things can be very ubiquitous. They contribute to observations where people are looking in the ultraviolet region, in the infrared region, present in a wide variety of objects in the solar system. And because of their ubiquity, there's also actually a hypothesis, which is called the PAH world hypothesis, the polycyclic aromatic hydrocarbon world hypothesis, which posits that these molecules, because they can form these very large scaffolds, might have provided environments or might have provided surfaces where other interesting reactions could have played out. But nobody has been, to date, provide a proof for how you can use such scaffolds as surfaces or templates to actually form molecules of an RNA world. No proof, but it's a very reasonable hypothesis that still exists in the field. This is just to show that their signatures are super prevalent, right? Reflection nebulae, planetary nebulae, AGB stars. You just see them in various intensities, but otherwise very readily present. PAHs are not the only molecules that are found, right? What are the other molecules? You can identify them using, scopes, but not enough because their vibrational and their vibrational transitions and all to identify them is completely non-trivial. So people take a combined approach. They actually do experiments in the lab. They look at the spectra that get generated from the lab PAHs, try to extrapolate it onto the data that they acquired from the various scopes to actually systematically think about, actually systematically assign the characteristic of the molecule. But here's that other meteorite I showed you, right? I talked about. It's called the Aguas-Zarcas CM2 carbonaceous chondroid. Same set of molecules. I mean, look at this column here. I will let you take a look at it for a second. In addition to some of the amino acids that we recognize as biological or proteinogenic present in our proteins, there's a whole bunch of them that our biology doesn't see. But they can form very readily under ISM conditions. And they can ride these rocks and get delivered to different parts of the universe, if you ask me. What are present? Nucleobases. This is I'm not going to go into the techniques that we use, but we extensively use a technique in biochemistry called high-performance liquid chromatography that allows us to separate molecules based on several properties. But you could find molecules that were relevant to the informational polymers, molecules that are relevant to the catalytic polymers, and sugars. All of this very readily possible in meteoritic samples. So there's this whole evolutionary story ahead of biochemistry that's played out on Earth much before we kind of appreciate the biochemistry that came about on our planet via terrestrial reactions. And these are more modern results of the same samples. What are these? These are actually AFM images. AFM stands for atomic force microscopy. It allows you to look literally at atomistic levels. And you can actually do lab experiments to generate the pHs, and you can understand their structures. And you can also draw out their, actual structures. Some of them they've been a little hard to assign, but most of it very readily possible. Just to say, interstellar medium chemistry, very important, but might have not been the only one. This is example from the Bennu sample. Again, a whole bunch of molecules that you recognize in biology today, and 14 out of the 20 amino acids we see in biology today, and all the 5 nucleobases of life, adenine, guanine, thymine, cytosine, uracil, all present. So why is life, as we understand it, only possible on Earth? Should that be the case? Something for you to think about. So I'm going to finish off by saying, I wanted to show you about how lipids do what they do and how they're formed and stuff, but hard to talk about so many things in one and a half hour session, but I'll show you this. Haji, abiotic synthesis leads to exact 50-50% formation of each handedness. Chemistry, just the chemical synthesis of a certain amino acid will lead to formation of 50% left and 50% right. Why biology chose what it chose is a fully open question. It's 50-50. Because abiotic synthesis has no preference for handedness. Where the enantiomeric excess happened, what processes sort of just allowed for that 1% excess of one-handedness over the other, which led to runaway effects later on, we don't know. I was just talking to Harish about it the other day. If the magnetic particles that he keeps talking about, there's actually a proposal that they might have induced some of this chirality, but there's no proof. It's a hypothesis. These molecules here on the right are what we work with in the lab. Tubes, circular cell-like stuff, lipids. This is actual sample from Murchison meteorite's core, which is only soluble in organic matter. And they form these same cell-like structures very, very readily. And more recently, not very recently, it's still already been a decade and a half now, when people looked at the composition of these molecules that sort of readily form these bubbles, these are actually hydrocarbons and fatty acids. Fatty acids, up to C12 fatty acids, have been found in Murchison meteorite. Suffice to say, your protocell-like boundary entities don't just exist in your labs. You can very readily find it actually in real-life samples. I'll just finish off with this by saying that when you're looking for, you come, buddy? Yes. When you're thinking about biosignatures and detection methods, there's a whole bunch of things going on, right? Scales all the way from atoms to few kilometers. His technosignatures have to be in the kilometer range, otherwise you're not going to be able to detect them. Atoms in the angstrom range, and there's a whole bunch of gadgets that can work across this size space to be able to detect molecules for you. He's touched upon some of the more specific signatures, so I'm not going to talk about it, but what I'm going to say is, sorry, this is just saying the signatures that these sort of concentrate a lot on. They could either be gases, pigments, some of the industrial sort of contributions he talked about. What are the pros and cons of them? Some are detectable, some are so unique, detection could be very difficult. Some are present in extremely small concentrations, making detection a problem. This is something he showed, so I'm not going to talk about it. But I want to leave you with this thought. These plumes are interesting, Ravi, and you should take them seriously because they talk about an underworld ocean. And in the whole crazy bunch of things that I didn't talk to you about, there's amazing biochemistry that shows that you can form those polymers, informational and catalytic polymers, in these conditions too, in sort of icy matrices. So hey, brackish water, very interesting if you ask me. So if we are going to be sending any more missions that are going to systematically look for habitability signs within our planet, I'll be hoping that they'll be sending landers to Europa and to Enceladus and not just flyby missions. We already have enough data from flyby missions. There's a whole bunch of missions that have already tried to understand astrobiological questions, and there's a subset of them that are actually in the works. But yeah, hopefully within my lifetime, maybe, just maybe this will be possible. Don't know, we'll see. But thank you for your time. Happy to take questions. I'm sorry I couldn't cover everything that I wish to. I think I'll leave you with that thought. Yes, Subbu. Yeah. Yeah.
You're in that sense replicating, but they're not evolving.
No, they're not.
Right? So is it possible to think of it in those terms? Because really it seems to me the leap that we want to make in life is to understand what is it that makes this particular thing which is going to become life, maybe not quite life, able to adapt. Right? So can we tie together things that easily form long chains with things that have adaptive capacity, though might not have all the facilities of life?
On paper, it sounds like a very interesting possibility. And there's this extremely small bunch of people, maybe the others are sort of hiding in the closet in this regard, are actually talking about how virus needs to be the fourth domain of life. However, going by the definition that we work with, virus doesn't make the cut because these guys are not self-sustaining. They are alive only in the context of a living entity. For that reason, I feel like viruses have not gotten enough of importance in the context of trying to understand if they might have been one of the early sort of steps. Oh, viroids. Okay, the RNA. Yeah. I feel like it's very possible. Actually, I'll take this leap to viruses and say, when you're talking about protocellular entities, viruses do make the cut. They have a membrane. They have an encapsulated genetic material. They actually also have some sort of minimal network of molecules that allows them to use the host machinery to be able to survive once they are present in that context. But the second you take them out of that, there's a problem. So to think about what you're saying, is there any way to couple some of the things that we are talking about or we are trying to simulate or make in the lab to some of these viroid-like entities? So one of the important things to remember, your hammerhead ribozyme is a viroid. It's a plant viroid. Am I comfortable stating that it could be an example of one of the molecules that came about in the RNA world? Absolutely. But unless and until there's a way to think about bringing it together with some sort of a minimal metabolic network and putting it in a membrane, the membrane could be a lipidic membrane or a proteinaceous membrane. I'm a bit agnostic to that. It's okay. It doesn't all have to be bilayered. But once you do that, it should be capable of embodying the three hallmarks that I talked to you about. Because of the presence of the RNA, it could replicate. Yeah, no problem. Should be able to do that. At least it can, even if it doesn't replicate its information, I'll take back what I said. It can catalyze its own cleavage. So it'll do that. Will it grow? Will it evolve or adapt? Do not know. Nobody has done these experiments systematically. But to talk about that circular RNA obelisks, I think the one problem we might run into is to think about how they can self-replicate themselves. Typically, when you think about self-replication in an RNA world space, you're thinking about a template, a primer that can go prime against the template, and then allow for further addition of molecules based on the information in the template. The second you think about a rounded off genetic molecule, if it's not large enough, I feel like steric constraints might come in the way for efficient replication. So you might have to invoke mechanisms that allow for it to open up, not lose its information while opening up, and then come back together. I think Shostak and his minions have shown a rolling circle kind of replication possible with a circular RNA. That's work that came out 3-4 years ago. But it's a little too, what do you call that? It's possible only in the lab. The templated replication, I would argue, should be possible even if Sudha is not present. If the template was present, the primer was present, and the activated imidazolides or other kinds of monomeric moieties that can attach were present, and there's somehow the dehydration phase is happening, Sudha doesn't have to do anything. Information transfer will happen. Possible. I have personally not veered towards it as yet. I do not know how many people in the field are using that as a way to look at least as a first pass, is it possible or not? I don't know, maybe people are doing it. I really don't know. At least I know that people that are working in the space of ancestral enzymes are beginning to use it because it's very easy to reconstitute, a hybrid enzyme or a synthetic enzyme using different parts of sort of pieces of peptides from individual proteins and come up with a new molecule. They're using it. I do not know if, for example, prebiotic chemists like me who work in the RNA world space are thinking about this. You just gave me an idea. Maybe I'll just do it. Yeah. Maybe you should send me the link if you remember or find it. Yeah. The protein folks are very happy about it. The peptide people are really working with it. Not as much the nucleic acid folks, I think. I'm not too sure. But at least I haven't veered towards it yet. So that is the other thing. You have a whole bunch of, you have a gigantic database for proteins in the space. You do not have enough for nucleic acids. Maybe that's why it's not happening. Huh, Shyama? Yeah. Right now, there's life there already. So let's say it, huh? You mean not happening on some other planet or some other body and stuff, or on Earth? Very possible. Yeah. On Earth, it is a bigger problem because there's so much life around, it's going to be very difficult to tease it apart. No, there's a problem because the conditions are not the same as what it was when all of these events were panning out 4 billion years ago. A lot of, particularly if you think about the terrestrial geothermal pools, the pools are not seeing the same kind of environments anymore. The atmosphere is not heavily, carbon dioxide, even though the volcanic outgassing is putting out carbon dioxide, or it's not necessarily volcanoes that we're looking at. We're looking at hot springs near geothermal activity. So things are more ambient. Even though we say Ladakh is a beautiful unlocked site and conditions are a bit extreme, it's not remotely comparable to what happened 4 billion years ago. So that would be something that will keep me worried about saying that maybe it's happening even today. But let's assume it's possible and it's happening today. The microbes that are going to be around there are just going to lap up everything that remotely looks like an energy molecule. It'll just chomp it off and use it up. Yeah. I think not. And I don't know the one that you're talking about. You can show me the reference later. I'll tell you what silicon is great for, to form sand. Beautiful reaction. Oxygen, and that's it. It will stay there. Sand, silicon, even though it's tetravalent like carbon, doesn't have the other properties that carbon is endowed with. Carbon can make and break bonds easily. Extremely important for chemistry. You cannot have one-way reactions where you're getting trapped and that's it, you cannot be used again. Silicon has that problem. So under certain other conditions, on some faraway distant world, where silicon's tetravalency is put to good use and reversible reactions are possible, can life be silicon-based? Anybody's guess. Yeah, silicon is. There. What kind of life would be possible? So the whole bunch of stuff that I didn't talk to you about, one of the things which I alluded to when I was answering one of his questions, when I was saying that we should take pre-biosignatures as something that's important. There's a whole bunch of alternate chemistries that can happen more readily than the chemistry that we see in biochemistry today. What's happening today evolved to be badass under ambient conditions. But very early on, there were other molecules that were even more, if I may say, superior in terms of their survivability under those harsher conditions. So do we have a better sense of these pre-biology driven understanding of worlds? Do you have pre-worlds that actually had a completely different sort of landscape of molecules? If yes, are these molecules something that you should keep in mind when you're actually sending out a mission to look for signatures? My answer would be yes, because they're much more readily plausible under certain conditions. Yeah. So, yeah, so silicon is great. Like I said, silicon is fantastic to make, irretractable solids that just form sand. So, carbon is amazing. It also makes glass. Oh, the experiment that she was talking about. Yes. So here's the problem, no? When we are talking about prebiotic chemistry, pre-biological molecules, they don't have any hard shell-like structures that can leave fossilized signatures. For fossils, there is a requirement for polymers, presence of polymers that can survive for extended periods of time. For example, bones. For example, chitin, a plant polymer that allows us to look at, fern kind of signatures and those kinds of things. When we're talking about these prebiotic chemistry molecules, like pre-RNA world molecules, for example, they don't leave any signatures. The only thing that we can try and do is to understand how they could have undergone crosstalk with molecules that are more readily sort of seen in extant biology to see how the transitions might have happened. Lekin kabhi unka signature hum pakad sakte hain? Can we ever catch one of these things? The answer is no, not possible. Very difficult. If it is a mineral signature, you can find it. If it's a mineral-based molecule, because zircons are minerals, right? Some of the oldest, proof for when water was present on Earth and so on and so forth comes from extremely old zircons. It even has signatures of the atmosphere of Earth. Geologists use this all the time to look for try and understand early Earth conditions. But in terms of the biochemistry or the prebiotic chemistry and fossils in that space, none. It's not a fossil in that sense, it's an isotope. So basically when you find those hot spring deposits, sinters, from which you extract some material, take it back to the lab, and you're trying to analyze the sample, you're looking for isotopic ratios that tell you whether your sample is from a biological source or an abiological source. So that is a molecular fossil in that sense. It's not in a classic sense of fossil in the paleontological sense. That happens. Yeah, that's what allowed us to find the 3.95-year-old signature. Predict. See, I'm so sorry. Now I understand what you're saying. Have they predicted it? People do simulations to understand how isotopes vary over timescales when certain processes are taken into account. That happens. But whether people have systematically tried to understand this from what are the potential signatures that we can look for if a certain reaction panned out under these conditions in a very systematic manner, I probably think I'm going to veer towards no as the answer. I think what you're saying seems very reasonable. People, have they done it? Has it been an interesting enough problem for them to look at it? I do not know. But thank you. I'll check and I'll let you know. I mean, I can let maybe Joe know and he can pass that information to you. Hi. Thank you. I'm glad I did. Okay. I think it's a unified sort of an approach. You cannot completely explain everything just by using laws of physics, because certain processes that happen in biology lie outside the realm of laws of physics. We still do not have a unifying principle that can explain it all. So I feel like you should just sort of wear a hat that's open to all possibilities and say that, oh, maybe this explains this, but we need to come up with something else to understand this other process, which is, yeah. Sorry, say that again. Complexity science, yes. And there's a lot of physics that underlies it. Yeah, possible. But there are people, okay, fine, sure. Sorry, said it to the wrong crowd. This would totally fly in a biology crowd. But no, so there's a lot of information science that people are using now to understand emergence of complexity and evolution of complexity. In that space, yeah. Yeah,
Maybe
I'll
one
take it
last
back.
question.
I shall take it back. Sorry.
Last question before call.
I think we're done mostly. Ah, yeah, Joe. Wet dry cycles, yeah. People haven't looked at it completely from a experimental perspective. There are people who have tried to do some modeling about this, but there's no necessarily experimental proof in this context as yet to say that, okay, this is a model, let's try and see what comes about, and then can we refine it and so on and so forth. What has been done is, at least in the microbiological space, microbiologists are interested in understanding how do you, beyond doubt, assign a certain snapshot that you get from your various missions as something that is clearly a contribution, that is clearly an outcome of biology versus something that is completely an outcome of abiotic processes. For example, these protocells that I'm talking to you about, when you look at them under the microscope, they look very cellular, cell-like. So how do you discriminate between a minimally not fully alive at all, right? Protocell is not fully alive, maybe a minimal living moiety. How do you separate it from a real cell which is living? This is something that more people are doing, approaching from a more systematic perspective, where both modelers and experimentalists are coming together, putting their heads to it and trying to understand, but nothing in the space of molecules. I think that's also because it's very hard, even let's say theoretically and experimentally, we figure out this some space which is conducive enough that will give us information and that will say, okay, let's go. If we find some old rocks, let's try and look for it. You will not find these molecules. They're super, what do you call that? They're very labile to hydrolysis. A lot of these molecules, if they get exposed to, water and stuff, they can undergo reverse hydrolysis very quickly under certain conditions. They don't need very high conditions also to undergo these back reactions and to break down. So they're not very, they don't survive for extended periods of time, which is why I think not much of time and effort has been invested in this space. The minimum complexity that is required for us to think about the question that you're asking about is at a level where things are already compartmentalized. Minimum, there's a boundary condition that allows things to sort of stay within a space, and maybe that still allows this to survive for longer periods. Don't know, but it's still not happening from a perspective of trying to understand rock records from early Earth. It's mostly being these attempts or these approaches are mostly being taken to understand what if we find a signature. Let's say Perseverance sends a picture to us from one of its cams, and it looks like a string of cells. How do you assign beyond doubt that it is biotic, so hence a microbial signature, or did it completely form because of abiotic means? I answered very tangentially, but that's the honest sort of picture of what's happening in the field now. I do not know. Maybe. No, can you once again? Planetary protection. It's not allowed. Come back samples. Huh. is coming back, no? Scrap, no? However, to answer one other aspect of something that you're alluding to, but not directly, if Mars has had microbes in its distant past, it's not there on the top anymore. That's for sure. We have signatures that we know that Mars has had water in its distant past. What people are interested in, and actually there are some gadgets on Perseverance, which is going to drill down. And in the subsurface, there is a reason to believe that maybe if there is any microbial thing that got frozen in time, that is where the signatures would be. But we are now not talking about molecules, we're actually talking about microbes, living entities. And they're doing, that was the plan. That was one of the sets of samples that was going to come back on the return mission. I think Perseverance is doing its job. Will it come back is the question. As of now, yeah, it's only in situ. But what Subbu says is interesting. You bring back a sample, you put it in water, in a petri or something, and then see if actually microbes come back. You said plant, I'll say microbes, if they come back. That's a very reasonable experiment to do. That's the problem. There's a whole bunch of things we're completely ignoring. This is like a thought experiment. Agar. And once it's come to a clean room and you're in a clean suit and everything is possible, then the question to ask is, will it now have, microbes can duplicate very quickly, replicate very quickly. So you provide the right ingredients, will it just come back? Very interesting experiment to think of doing.
So I think on this note, let us thank Sudha again for a very nice talk and discussion.
Thank you.